Once upon a time, there was a brave strawberry warrior named Fragaria Anassa, or Frag for short. He was gravely injured after a harsh battle against the insects, because he was grown organically, so he didn't have GMOs to protect him. Although he is injured, he must endure the pain and travel back to his hometown, Strawberry Land. On his way back home, he suddenly encounters a peculiar organism. Upon closer inspection, Strawberry realizes it's the legendary crop killer, Grey Mold.
Oh no! The crop killer is right in front of me. I must defend myself.
Mwahaha, you have very bad luck, for you have encountered me, the one and only Grey Mold. You may have heard of me by my scientific name, Botrytis cinerea.
Yes, I have heard of you. You have been notorious for causing extreme pre- and post-harvest diseases in over 200 different plant species. You're a mass murderer. I'm not worried though. I can just run away from you.
That is where you're wrong, silly Strawberry. You are a plant and realistically cannot move. I am a fungal pathogen that can travel and spread airborne. Through this method, I've been able to easily spread and cause outrageous damage to multiple plant species, including grapevines, tomatoes, cucumbers, and other strawberries like yourself. I am a necrotroph, meaning that I will feed on your dead cells.
The Grey Mold was right. Botrytis cinerea are capable of spreading through the air. The initial cycle begins when Grey Mold lands on a crop in the form of a conidium, a spore that is produced asexually from the Grey Mold hyphae. The Grey Mold begins its attack by landing onto the strawberry's surface.
Aha! I have now successfully attached onto you.
No, I was careless, but not all hope is lost. The next step of infection is the germination of the germ tube. I know that various factors have an influence on Grey Mold germination. One factor is moisture, and another is ethylene. Right now, it is too dry for you to do anything.
Frag the strawberry is correct. Germination of the germ tube is a necessary initial step for penetration on the plant cell. However, luck does not seem to be on his side, for suddenly it began raining.
Mwahaha! Now I could begin my germination process.
Oh darn! I also forgot that I'm an old strawberry, so I'm producing a lot of ethylene. Luck is truly not on my side.
The hydration from the rain causes weak adhesive forces from the hydrophobic interactions to form between the conidium and strawberry, allowing it to stick better. History, in other words experiments, has shown that germination occurs more efficiently in moist environments. Ethylene has also been shown to elongate the germ tube, allowing it to invade the host more efficiently.
Haha, now that my germination process is almost complete, now I can form aprosoria.
I don't remember what that is. What's aprosoria?
Haha, silly fool. Aprosoria formation is the next stage of my developmental cycle that forms from the germ tube. It is essentially the formation of specialized cells from within me, ultimately contributing to the swelling of hyphal tips, increasing water absorption to stay hydrated. Aprosoria is also what I use to ultimately penetrate through host tissue with the help of membrane protein tetraspanin.
Grey Mold has begun its penetration using aprosoria into the host's surface through the method of passive ingress at Frag's wound site from his previous battle.
Oh no, if only I didn't get this cut during my fight against the insects.
You truly are a fool, Strawberry. Whether you had the injury or not, it would have made no difference. You see, there are two possible methods for me to penetrate the host tissue. The method I am using on you now is called passive ingress, which allows my aprosorium to enter through open wounds. The second method that could have been used is through active penetration, where I can utilize enzymes such as 6-D-KDA lipase to help germ tubes of the conidia to enter the tissue. This is important because it allows for initial entry into the tissue.
Experimental studies have shown that when lipase is inactive, germ tubes have difficulty getting inside the tissues. Thus, this suggests that lipase plays a regulating role to help break the cuticle.
Oh no, this is the end for me.
It is indeed. It is time for me to destroy your cells.
The Grey Mold has various methods of damaging host cells. One of the methods is expelling phytotoxins such as botrydile, which induces chlorosis and cell collapse. A second method is induction of reactive oxygen species, which leads to oxidative stress in plants. This could also ultimately lead to programmed cell death through oxidative burst by the host itself. Finally, fungal pectinases, enzymes encoded by multigenic families, help degrade the cell wall of the host cell by specifically degrading pectin. As the cell walls are degraded, tissue maceration occurs.
I guess my time is almost up. I can feel my life slowly fading away. Why do you do this to me?
This is necessary to sustain my life. The cell degradation is what provides the nutrients for me. Thus, your demise is necessary for my survival. Now that I have actively degraded your cells and that your tissue is macerated, I can undergo sporulation to create more conidia.
The gray mold aprosorium will spread throughout to other cells. The aprosorium will give rise to conidophores, which are conidium-bearing filaments. These will form even more conidia. This is the fungal biomass that can be seen on the strawberry.
It's a good thing I'm not near any other strawberries, or else you could easily spread to everyone else and cause significant damage to the environment.
Unfortunately for you, before encountering you, I had passed by Strawberry Land already. Many of my conidia spores have already infected the strawberry population in Strawberry Land. Right now, the only survivors are immature strawberries, but that is only because gray mold are known to have a special ability called quiescence, which allows me to stay dormant up to several weeks. No symptoms are showing on those strawberries because my spores are laying dormant, but soon they will meet the same fate as you.
No, I don't believe you. My whole village is destroyed. Why would you want to stay dormant in the immature strawberries anyway?
I'm surprised you don't know this. Because you're a strawberry after all, immature strawberries are known to have high levels of antifungal compounds that decrease as they ripen. Quiescence is my secret ability to avoid this host defense response.
As gray mold has said, young immature fruits contain high levels of antifungal compounds known as phytoalexins, which decrease as the fruit ripens. In order to avoid this host defense response, gray mold undergoes quiescence, which allows it to stay dormant in the plant until the fruit ripens more before symptoms begin to show.
I guess it's over for me and my village. Strawberry Land is doomed, and so am I. Oh no, Mr. Greymold, I suddenly don't feel so good.
Frag the strawberry is now experiencing the symptoms of a full infection of botrytis cinerea, including soft rotting of parenchyma tissues and an abundance of conidia on the fruit.
Farewell, Strawberry. Through this process, I hope you had a great educational experience as I was infecting you. If you could still respond, let me know if you have any questions. For now, I will now fade away, spread out, and continue in my conquest of infection.
In summary, the conidiophore from the hyphae released a conidium spore and begins the germination process, forming the tube for a prosorium. What occurred in this story was that the conidium of the botrytis cinerea attached onto the strawberry. It began germinating, and eventually it penetrates into the strawberry's tissues. The beginning of cell degradation within the strawberry consequently begins, and it absorbs its nutrients and begins to expand into the gray mold biomass. The cycle then repeats.